Welcome to Genialis expressions powering Diagenode CAT's RNA-seq data analysis. You've prepared sequencing libraries using Diagenode's CATS kit, and finally have your data in hand. Time to dive right in. Genialis expressions provides tailored bioinformatics pipelines and single quick analysis of the raw data, and makes downstream data exploration easy, fun, and rewarding. Let's get started. Sign in with your Genialis account, or if you do not yet have one, simply register for one here. Once signed in, you arrive at your personal homepage full of helpful hints, tutorials, and documentation. Your most recently analyzed data is linked conveniently on the homepage as well. First things first, let's upload your data and trigger the analysis. This is done on the recipes page found here in the navigation bar. To start, define your experiment. You need to specify the organism and choose the analysis type. Then, upload your FASTQ files via drag-drop or clickable file browser. While your samples are climbing on board, let's double-check that any mate pairing was done properly. You may also edit the sample names to something more user-friendly. We recommend unique, yet succinct sample names. Once the sample files are uploaded, start the analysis by clicking the big blue Run button. With a single click, the automated bioinformatics workflow will perform quality control, CAT-specific trimming, alignment, and quantification of gene expressions. Next, we come to the Search and View Results page, where you find analyzed data, new and old. Hover over or click the status icon to see a real-time progress report. The results table populates as the analysis runs. Everything in this table is clickable. For example, you can check the quality of your data by clicking on the QC report link. Clicking on the sample name takes you to the sample details page, with all the gory details of the bioinformatics processing and a sample annotation form from metadata. While we do not want you to sweat the bioinformatics, we don't believe in black boxes either. Now for the good stuff, data exploration. For the purpose of this tutorial, we have pre-populated a small dataset prepared by Diagenode to analyze the effects of the drug MS-275 on liver cancer cells HepG2. First, find the samples of interest. I have selected two sets of samples prepared from HepG2 cells and from secreted extracellular vesicles. I can add these samples to the sample basket, just like shopping on Amazon. Okay, time for the good stuff. On to the visualizations. We find 5 dashboards or views with which to answer certain questions. My first questions always are, did my experiment work? Are my replicates consistent? We can see that the replicates form clades as expected, and some distance between sample types, consistent with the distinct transcriptome profiles. The drug MS-275 is a histone deacetylase inhibitor. So let us next observe how various HDAC genes are expressed. You may search for a gene by its symbol, or paste a list of genes here. We see HDAC expression ranges widely across cellular and secreted samples. A next logical step might be to check which genes are differentially expressed between drug treatment and control. You can trigger a new differential expression analysis by clicking the Create New button. I'm selecting a previously computed analysis. In the volcano plot, we can see the genes that are differentially expressed and draw a box to select those downregulated in response to MS-275. I can add them to the genes card, as well as save a new gene set for later analysis. I want to know which of the genes downregulated in the cells display increased abundance in the secreted extracellular vesicles. To do this, we will be using the Venn view dashboard. Let us create a new Venn diagram with the different upregulated and downregulated gene sets that I have previously saved. We see that the 3 of the genes downregulated in the treated HepG2 cells are upregulated in secreted extracellular vesicles. Now, let us find out if any of the genes downregulated in treated HepG2 cells show striking visual patterns that might suggest drug sensitivity. For this, I'll use the Heatmap dashboard. I've selected only the HepG2 cell samples. I have also log transformed and clustered the data to visualize groups of similarly expressed genes. Aha! Some of the genes, like IL-17RB and TYMS, are known to be involved in some cancers. This may be worth further investigation. At each stage of your analysis, you can export the data tables and plots using the export icon here. If you want to further analyze your data on your local machine, you can do so by selecting the samples you want to download and then the files you want to download. You might also want to share your data with your collaborators. We support granular permissions on all data. As long as your collaborator has a Genialis account, you can share the data with them. This is Genialis Expressions powering Diagenode Cat's RNA-seq data analysis.